Hello, everyone. My name is Haeree Park, and I spent this past summer in Dr. Bruce Walczak's lab, where we looked into a canine cancer immunotherapy option utilizing NK cells and tumor-targeting monoclonal antibodies. So just to give a brief overview, we know there are very few clinical applications of canine immunotherapies to treat canine cancers in veterinary medicine, and the research field itself is still not well established. However, in human medicine, there are human immunotherapies that are extensively researched and applied commonly in cancer therapy for human patients every day. And so we wanted to use a specific human immunotherapy model utilizing NK cells or natural killer cells to target canine malignancies. And so my research is really focused on this portion of this figure where we have an NK cell interacting with a tumor cell with the NK cell's FC receptor and an anti-tumor antibody that is binding to a tumor antigen, causing what's called ADCC. And so we want to use this same concept in our canine patients. And so what is ADCC? And so ADCC is antibody-dependent cell-mediated cytotoxicity. And so what happens is there's an NK cell with an FC receptor, and this FC receptor recognizes and binds to an anti-tumor monoclonal antibody. And this monoclonal antibody recognizes and binds to tumor antigen on the surface of a tumor cell. And so in my experiment, we are focused on two FC receptors. And so they are called Canine CD16 and Canine CD64. And so these are canine FC receptors. However, they are on human NK cells. And so we had to recombinantly make this in a previous study not shown in this presentation, but these were created. So this canine FC receptor recognizes a canine anti-tumor monoclonal antibody called canine cetuximab. And so this canine cetuximab recognizes EGFR1, which is an epidermal growth factor receptor, and this is highly expressed on tumor cells in general. And so when this complex and interaction occurs, you have this NK cell that is supposed to induce cytolytic factors and anti-tumor cytokines and cause the release of those and cause the eventual death of these tumor cells. And so again, these NK cells are human in origin with a canine FC receptor. And so the human NK cells are called NK92 cells, and these are our effector cells. And the tumor cells we used are a human ovarian cancer cell line called SCOV3, and these are our target cells. So you see different E to T ratios, effector to target ratios of how many NK cells do we put in with one tumor cell. And so putting this into action, we wanted to see with these two FC receptors of interest, Canine CD64 and Canine CD16 shown on the x-axis, at different effector to target, at this effector to target ratio of 10 to 1, so 10 NK cells to every one tumor cell, we wanted to measure how much tumor killing there was as percent lysis, or how much overall tumor cell killing there was in the presence of canine cetuximab, that anti-tumor monoclonal antibody. And so what we see is that this NK cell with canine CD64 as its FC receptor, we have pretty high amounts of percent lysis, or tumor killing, by this ADCC mechanism of action. However, canine CD16 didn't seem to show much tumor cell killing at all. And so moving forward with canine CD64 as a promising option for an immunotherapy, we wanted to see what canine CD64 does in relation to how it would work with canine cetuximab. So again, on the right, you see canine cetuximab in the presence of it or not at different effector to target ratios. So we have human NK cells with this canine CD64 receptor, and we show at different effector to target ratios. At all of these ratios, we have a good amount of tumor cell killing shown on the y-axis here. In comparison to how in a natural setting with human CD64 Fc receptors on human NK cells with a human anti-tumor antibody, how is it supposed to work with, let's say, something that we know works in the human model? We see different effector to target ratios in the presence of cetuximab or not. We show that it has a very similar profile with the percent of lysis and percent of tumor killing, which pretty much tells us that this canine CD64 Fc receptor does seem to induce ADCC with our tumor cells of interest. And so in conclusion, just to summarize quickly, we show that these engineered canine NK cells expressing canine CD64 can provide an adoptive therapy. So we'd have to swap out that human NK cell portion with a canine NK cell, but using that canine CD64 Fc receptor. And then our next steps would be to kind of see what the expression of these two Fc receptors, CD16 and CD64, are in these canine peripheral blood or PB NK cells instead of using the human NK cell line that we've been using. Thank you so much for your attention.